spread around out in the main lobby. So now it is my pleasure to introduce Yuru Chen, who will be introducing our first speaker.
Hey, good morning, everyone. I'm Yuru Chen, a PhD student in plant breeding program at ISU, and I work with Dr. Thomas Ruberstadt in maize haploid inducer research. Today, our first guest speaker, Dr. Marcio Resende, is the assistant professor of University of Florida, and he got his master's degree from Federal University of Viçosa in Brazil, and the PhD degree from University of Florida in genetics and genomics. Now, Dr. Marcio Resende is the leader of the corn genomics and breeding lab UF. They are helping the Florida farmers to maintain the steady in sweet corn production. Okay, so today his speech is to talk about the genomics and phenomics applied to sweet corn breeding. Okay, so please help me welcome Dr. Marcio Resende.
Thank you, Yuru. Let me see if that's going to work. Can you guys? All right, good morning, everyone. Really, thank you for the introduction. Thank you for the invitation. Really happy to be here. Just to echo a little bit what Dr. Lange said, thank you, Corteva, for organizing these events. I think it's an amazing opportunity to network and to have the students organizing this, and great job for all the students that put this together this year. It's my first one of the series. I am a faculty advisor of the group in Florida, so I go through all the stress, all the organization. I actually don't do much. They organize it all themselves. But great job. And I do want to start saying that when Yuru asked for a title, I wanted to keep it broad to have an opportunity to talk about what we're doing in sweet corn breeding. And I said genomics and phenomics apply to sweet corn breeding. I do hope that none of you woke up early and came hoping to see some very fancy phenomics. And really, I think you're going to see that from Chris and others later on. But we're mostly talking about how we're setting up the program to combine phenotypes that we're collecting with genomic data and really try to move the program forward. The program in Florida is a sweet corn breeding program. It's an inbred development program, and I'll talk a little bit more about that. But most of you here know about sweet corn, but if someone doesn't, and it's someone that's not familiar with it, technically speaking, the difference of sweet corn compared with field corn is basically mutations in genes or knockout enzymes in genes of the starch biosynthesis pathway that therefore increase sugar content in the endosperm. And commercially, the two genes that are used are shrunken 2, which is encodes for cytosolic AGPase, and sugary 1, which is down here, is an isoamylase. And this figure actually comes from Al Myers here from ISU. So you will hear me talking about these two genes throughout the presentation. Fresh market sweet corn nowadays, almost 100% of it is mutants. So that's the AGPase mutants. So what that leads to is if you buy your corn in the supermarket and you let it dry, you will get these seeds. You'll see these ears. They're really shrunken and shriveled because they don't have a lot of starch and they lose all the and the ears look really ugly. We actually are, when we're planting, sometimes we look at them and say, this is never going to germinate. And then incredibly it does. But in reality, while this is technically the only difference between field corn and sweet corn, there are several other requirement traits that breeders over time have been selecting for, and which many of you will know, the sweet corn hybrid looks actually very different from a field corn hybrid. So yield for us, and I'm speaking strictly about fresh market, which is what we do in Florida. There's Bill Tracy has a breeding program in Wisconsin for processing. Yield for us is marketable yield. We actually measure crates per acre. And what that means is an ear that doesn't fit all the way to the top, it's not a marketable ear. And most of the harvest is actually a manual harvest. So some traits that are important for us are, for example, ear snap. How easily can you actually get the ear out of the plant? The plants cannot be too tall, and that's just a logistics of how the harvest is done. So plants are generally a lot shorter. The hybrids are a lot shorter than the field corn hybrids. And then there are a lot of aesthetic traits. So we care about colors. We care about how dark are the husk colors. And the silks cannot be red because especially in the processing market, if that goes into the can, it looks like a hair, and it's not really a trait that we want. So there are all these little requirements that first make the breeding quite challenging from a point of view of, for example, if we want to increase yield, the ears cannot be too long because if they don't fit in the crate, they're not. They're basically tossed. And then, like I said, for example, sweet corn has these what we're called flag leaves, but I'm referring to the ear flag leaves coming out of the ears. And you see those in the supermarket. We don't ever see that, or we don't really see that in dent corn. It's purely aesthetics, but it's something that the consumer wants to basically gives a sign of to see those little ears coming out of those little leaves coming out of the ear. So there are a lot of these constraints that we need to keep an eye on. And in addition to that, so these are the traits we select for, besides insect resistance, disease resistance. But in that, there are just some traits that are naturally different. And again, the plants are generally shorter. Has anyone seen the tassel size of sweet corn hybrids? They tend to be a lot bushier and bigger. And I will actually come back to that a little bit later in the presentation. Most of the materials are much earlier flowering. We generally do have somewhat of a poor germination, and stem counts are key traits for us because the seeds have a lot less reserves. And the other one you often see corn plant with like open leaves, and leaf angle has really not been a trait that breeders have selected for over the years. So just to say that there is a lot more than just starch mutant genes that go on into these hybrids. And I will come back to specifically talk about tassel size. So how big is the industry? It is a specialty crop. It doesn't get anywhere close to field corn. But nonetheless, it is one of the most popular vegetables in the United States. Actually, consumption comes third right after tomatoes and potatoes. So we do eat a lot of corn. Florida ranks #1 nationally in the production and value of fresh market sweet corn. That's why they have a sweet corn breeder there. Acreage-wise, we plant about the same amount of silage corn and sweet corn. Value of sweet corn is much higher because it's a specialty. And it is our 5th most valuable fruit and vegetable crop. The market is about 200 million. Just to put in perspective, we plant about 30,000 acres per year of sweet corn. Iowa does, what, 10, 15 million of field corn? So acreage-wise, it is a small industry, but it is high value, it is a specialty value. And the total value for the crop is about $1 billion. And in Florida, like most of the fruits and vegetables, we try to capitalize on the window, on the early window, and get these produced through the winter. So the count is most of the production is in South Florida, which just from a point of view of me based in Gainesville, Florida, which is here up north, it means about a 4-hour drive every time I need to go to the field, which is somewhat of an interesting challenge, especially during peaks. But anyways, most of the production is really nearby Miami, and Palm Beach County is where most of the production is. So we're focused on inbred development. And so our model is to develop inbreds, partner with the companies, put these inbreds through test crosses, and then license the inbreds to companies, and then they produce the hybrids. So we actually do have right now 2 commercial hybrids that are in the market that use our inbreds. And I came on board. These were developed by my predecessor. I came on board 3 years ago, and that's one of my main roles, is to just carry what I'm calling an operational breeding program, a field breeding program. And then on the side, and what I'm going to be talking most today, is this genomic work that's going on to hopefully allow us to translate some of these new developments to the operational program and help speed up some of the developments. Production generally is growing, which is good. So something that may be interesting for you guys who do a lot of corn here in the Midwest. We can plant as early as January. Early in the year, the South Florida is actually fairly mild winters. So as of right now, we're doing, we can do 3 cycles a year. It gets a bit crazy. Have to have people working through the holidays. So we actually decided not to do that this year. So we basically have 4 seasons, but they are a bit overlapping. But there is some logic to the madness there, where we evaluate the years in South Florida and we decide the crosses based on that in North Florida. So we're planting again, we plant in a few weeks, and then we send these materials to Idaho, where all seed production for seed corn happens. So then these have to go and be tested for adaptation in Idaho, and there's a fall season. So it's basically a lot of field work that keeps going. One of the aspects that I was asked to talk about today was introducing varieties to new environments, and I think just briefly touch on it, but there is a significant amount of G/E from the locations in Central Florida to the locations in South Florida. Believe it or not, the soils are completely different. If you think about Florida, we usually think about sandy soils, and actually Palm Beach County is right below the Okeechobee Lake, so there's a lot of organic matter, and there's really a dark soil that's completely different. So it also poses a challenge from the point of view of selection. And throughout my career, for the past, 12 years or so, I've been working quite a bit with genomic selection. I've been preaching about how genomic selection works, evaluating in different species. So really, when I came to this position, my goal was to, among other things, to really implement it and figure out a way to make it, to actually use it and move it beyond just papers and publications. And that's what we're trying to do now, and I won't have too many results to show you yet, but I'll show just how we're setting up to get that done. And I believe many of you here will be familiar, but just if you're not, the idea of genomic selection is collecting phenotypes of interest, breeding targets, whatever they are, and calibrating statistical models where we use DNA information to predict the phenotype. And the advantage of that is that we can then go to early prediction, or we can just increase the intensity, or I can screen my plants in North Florida, extract DNA, and predict how they'll behave in South Florida, and really allows to speed up the cycle and so on. So just an example of that, and we did this recently in field corn, actually, in collaboration with a company, where we analyzed data from 230 inbred lines from field corn. So they were phenotyped, so different crop schemes, and we had a total of about 3,800 hybrids that were phenotyped in 18 environments throughout 3 years. And we did a series of evaluations for different models, for jointly modeling environments and over the years. And in summary, so this is going to come out soon in crop science, but in summary, the model that worked the best was a multi-environment model that considered all the information at once, and actually adding non-additive effects actually did increase the accuracies, and they performed fairly well. Now, there are several other groups and several other publications that have shown results similar to this, but the reason why I like to point this out is because the company that was evaluating this project went on to really convince senior management, and they got 3 breeders to select which hybrids to be planted given the same pool of inbreds. So 3 breeders did their best selections. One of these breeders used genomic selection models. The other 2 used their conventional selection tool set that they were used to do. And then they planted this trial side by side. So you had basically three hybrid trials side by side from the same pool of inbreds, from the same pool of hybrids. And when they then, and I'm just showing here relative yield relative to the lowest yield, when they ranked the yield of the top 50 hybrids based on who selected those hybrids, the two trials that were in the same environment that were selected from genomic selection models, they had the highest yields. And that really resonated with senior management for whether it was worth the investment on genotyping. Not only that, this trial was evaluating a total of 1,000 hybrids, out of which only about 25% of them came from genomic selection. And we actually looked at the top 20. Out of those top 20, 12 of them came from the predictions. Towards everything I'm going to show you today are coming from USDA NIFA Specialty Crop Research Initiative grant, an SQRI grant, specifically to improve sweet corn. And I just wanted to then acknowledge them up front. Thomas Luberstein, one of the co-PIs, and there are several other institutions. UF is a leading institution. We have USDA, Wisconsin, Washington State, and then Thomas Anushi here in Iowa. But nonetheless, this is a line that has a shrunken mutation, which is what's mostly used nowadays in fresh market commercial sweet corn. And a line that's it's an Iowa 4.3 line, shrunken 2. And the reason why we did it is a public material, but it has significant presence in many of the commercial materials out there. So we felt like we're at least representing well what's out there. And just to walk you through a little bit of the process, if you have more questions, I won't go into too many details, but I'll be glad to answer more questions. This line was sequenced with PacBio, so it was sequenced with long reads. We collected about 65x coverage, so each read was sequenced on average about 65 times. Each base pair was sequenced on average about 65 times. And we went through a pipeline for assembly using Canoe and bringing different tools that help the assembly. So Corteva helped us with the BioNano optical mapping. Initially, we actually went with dovetail high C mapping for the scaffolding without BioNano, and that didn't work too well. So we went back to BioNano and combined them. And then finally, at the end, brought Illumina sequences for errors and ended up with an assembly that was very good. We have more than 90% of the genomes in 10 scaffolds, which represent the 10 chromosomes. Total length of the genome was about 2.3 gigs, so a little bit bigger than B73. And the longest scaffold is basically a whole chromosome, and it's about 304 megabases. So that basically the starting point. And we did a few more comparisons to look at the quality of the genome. And these were compared against the version 4, B73, where we're looking at how much genes that are generally conserved, how much of them were fully assembled in our assembly. And the results are comparable and often better than B73 version 4. So we were really happy that this process ended up with a genome that we think that really will have an impact for sweet corn. I won't talk much about CRISPR today, but there's a few projects going on with design of guide RNAs and gene editing, and that was also the other reason why we wanted to have a genome, because there's a fair amount of polymorphism between sweet corn and food corn. So a genome is no good without an annotation. We went through different pipelines and ended up settling on an annotation that basically used, it's a pretty standard process that used Maker, and then gene predictions using FGNesh and Augustus. And then we did use other field corn annotations, the ones that were available at the time, combined that with RNA-seq from our own genome, and then created a final gene set that resulted in. So at the end, we have 38,000 transcripts after some filtering that have been annotated. And as it's common, but it's just mind-boggling to me, is just the fact that about 1.6 gigs of the genome transposable elements. So that's just how it is, right? So my student and postdoc, Vincent Colantonio is a graduate student working on this, and Ying Hu is my postdoc who is working on the assembly. They really like circus. And that just shows a little bit of the comparison of our genome with B73. So we found about 1 million SNPs, which is the red track here, well distributed in the genome. We found several presence and absence variants, large structural variants. So really, it is a very different genome. And that's just, again, for us, it showed that it was worth it doing, and we were not just replicating what genome that was already available. So just an example of some of the things we're getting out of this is, I mentioned to you SHRNKN2. So SHRNKN2 is of the SHRNKN2 gene. So all commercial sweet corn, SHRNKN2 sweet corn, has the same allele, basically. And for several years, researchers tried to figure out what kind of rearrangement actually happened with SHRNKN2. And they were not able to do so using conventional methods, mainly because with this assembly, we were able to find out that there is a 50 kb insertion. There's actually a threefold rearrangement. So this is the gene in B73. A piece of it got inverted. There is an LTR insertion right here that's about 5 kb. And there is a 50 kb insertion in between these two pieces. And that includes mainly a helitron, but also an LTR insertion. And this region is highly repetitive in the, so any other approach that was used in the past to try to recreate what was going on didn't work well. So we knew that there was a rearrangement, but we didn't really know what was the final structure of the gene. And the reason why it is a knockout is there is gene expression coming out of it, but when we look at the transcript, it breaks in the main domain, which is the glucose 1-phosphate adenyltransferase. And that's what really knocks out the gene. And then just another example of something that we got out of the genome is we looked in these presence and absence variants for things that were unique to IO453 compared to the other genomes that were available at the time. So that's really going window by window and checking whether a given sequence of the genome is present or not, and whether there's any gene annotated within that region. And basically that becomes defined as an IO453 specific gene. Then the question is this a sweet corn specific, or is this just a line specific? And one of the things we found so far, and then there are more of these, but just one example is a sphingosine kinase that is basically it's a 3 kb insertion It's present in IO453 and absent in B73, in MO17, in W22. So we actually downloaded some RNA-seq data of AIMS222, and it actually seems to be a sweet corn specific gene expressed in the endosperm, in the kernel, whole seed. So we have here 5 sweet corns, and they all have gene expression associated with it. And the field corns that we've looked here had no expression. Sphingosine kinases are usually associated with ABA response. We're doing some tests whether it has anything to do with germination, which is a trait that is different between sweet corn and field corn. The exact function and reason for this is yet to be discovered. So that was a story of the genome, and then moving on to a second that we wanted to explore the genomics of sweet corn to really understand how to move forward with the program was to see where sweet corn in the whole scheme of things of corn. So we had a lot of public available GBS data, and we built a phylogeny merging all the field corn that was available with sweet corn genotyping data. And there was a series of filterings for the SNP set to reduce things, remove things that had a lot of missing, things that were in LD with each other. But we ended up with a phylogeny that's quite interesting. Not too surprising, but interesting nonetheless. It is rooted in teosinte. The first versions, you have central Mexico lines, northern Mexico lines. We have US land races and Pueblo land races. And then most of the sweet corn materials, pretty much all of them are in a single clade right next to what I'll show in a minute. These are flint lines. So that just supported the expectation that most of these lines were derived from northern flints. And going on, we have popcorn, and then we have a new clade of Central America and South America, several of the tropical lines, and then here we have stock materials. Some of the things that are interesting coming from that is a few years ago, Bill Tracy and Ed Buckler reported that the sugary 1 gene, the other gene that I mentioned to you that's commercially used in sweet corn, that there are 5 natural mutations for sugary 1. So throughout history, 5 times that gene got mutated and kept selected for. And we went back to the accessions that have the different mutations, and we tried to place them in this tree. So while the majority of the sweet corn is in here, we have 2 maize dulce land races are clustering with the Mexico group, and that's a 1.3 kb transposon insertion into the sugary gene. There is a Hotavilla land race that was thought to be putative clade before. We knew where that was going to go to the southwestern 12 rows, and if I go back, you see that this is where the US Southwest was. Nueta was reported to be derived from Great Plains flints. And again, here we have F7 and AP1, 2 flint lines that were clustered together, and that is indeed where most of the flints are grouping together. And then pretty much northern flints is the other mutation in sugary 1, and that's where all the other clade is. So really, commercial models are exploring the northern flint mutations and selected for that, which poses a significant bottleneck. All sweet corn was initially coming from materials that had that allele. So, and we have a tropical sweet line that is not very well known, and we actually don't know exactly. We're sequencing this, but we don't know exactly which mutation it has. Now, some of the other cool things we've done with it was then to get AIMS282 panel, remove everything that was sweet, so keep the non-sweet AIMS282, and compare signatures of selections. So I went back to the beginning of my talk. I told you that sweet corn plants are very different than field corn. So we wanted to find out what's different in the genome, what's been under selection. And we've done that through a variety of ways, looking at Tajima's D, looking at nucleotide diversity, and as we would expect, chromosome 4, where sugary 1 is. So I'm showing here nucleotide diversity in red for AIMS282, and in green for all the sugary 1 materials. So we have a strong evidence of a selection sweep there, as you would expect. That's not too surprising. We see the same thing for for shrunken 2, where shrunken 2 is located. But that was just served as a confirmation that at least we were capturing some biology with the analysis. But there's a lot of candidate genes in here that have not really been reported under selection from sweet corn to field corn. So we have a lot of early flowering genes. So that's combining the Tajima Z with XPCLR, which is another measurement of whether a region can be putatively under selection or not. So we have a lot of flowering genes. We have some sugar transporters. And interestingly, one of the ones that was the highest evidence of selection was the DPF2, which I went to search for what that means, and I didn't know, and I found this paper where the GWAS hit, quite a significant hit, confidently linking the ZPF2 gene to tassel branch length. So whether that's the case or not, I don't know, but it's quite interesting that we have differential selection there for a gene that potentially could be explaining why sweet corn have much bigger tassels. I don't think breeders have selected them to be bigger. I mean, we know that field corn breeders have selected them to be smaller, so it's one way or the other. So that leads to several candidates, and this is more of the non-applied part of my group that will be doing some validation and testing whether these things are real or not, but they could indeed have applied implications. Sweet corn has low diversity, and some of the things that we've talked about is bringing field corn as a source of diversity. If we know some of these alleles are important, and if we identify things, for example, for pericarp thickness, if you get a field corn and you make it sweet, unless you change the thickness of the pericarp, that corn will not taste good. So if we identify these things, they then become actually applicable to the breeding program. So the next step then, what we're doing right now is to get all the sweet corns in this tree, and we have about 700 of them, and we're sequencing, whole genome sequencing all of them, about 10x sequencing. And I have very brief preliminary data for that. We have about 1.5x coverage sequence so far. This is literally coming right now. And we have identified about 30 million SNPs only within sweet. So that aligned against our reference genome. So this really eventually will lead to the HAP map that will allow us to develop these markers for genomic selection. Interestingly, and this I generated this yesterday, so this is really just a visual analysis right now at this point. But chromosome 4 is visually has lower SNP density, supporting the low nucleotide diversity that we've seen. So that's a lot of what we've done in the genomic side. And I have a few more minutes that I just wanted to then explain which traits we're collecting and how we're approaching the data collection for that to then combine the two things in the future. And that goes back to the SweetCap project. It's a coordinated agriculture project. It's a collaboration of multiple PIs. And more in Florida, the growers want to plant as early as possible, and that's to fit basically the production window that no one else can produce. So I was mentioning to someone that usually we used to plant, a few years ago, the bulk of the planting was generally February for commercial sweet corn planting. It's now pushing to January and even December in some cases. So cold germination is an important trait for us. We can get the plants out of the ground when the soils are cold. That would be an important trait for that. And the way we're approaching this is all this population, all this diversity population was sent to Washington. And Dr. Lindsay Dutoit at the University of Washington is screening it for tolerance and basically planting there in a window where the temperatures are much colder than Florida. And at this point, just for stand counts and how fast and how much they come out of the ground. And then Mark Settle's lab is actually doing trial, and that phenotyping trials where you actually take pictures and image them and collect more quantitative data on when and how much they come out of the ground. But just to have a little bit of breeding comparison of lines, I know most of what I'm talking is genomics, but we have already identified several lines, for example, that repeatedly, and this is just one of the trials, they have much higher emergence in cold soils compared to the standard check. These are actually inbred lines. This was a hybrid. We do have an inbred check as well. But this is looking promising. Eventually, we hope to then have a genomic selection model that combine the two things, and I potentially would also have some candidate genes to lead to either markers of selection or gene editing, depending on the effect. So flavor is an interesting story. Flavor is something that I really like, and I've done some work with tomato flavor in the past. Breeders do screen sweet corn for flavor, but the reality is that there's not really a model where the grower is rewarded by flavor. So yield is always number one, because if you have a good tasting hybrid that doesn't yield the most possible, that's not good enough. Nonetheless, there are significant differences, and we walk the field and we do what's called a biopsy, and we just bite these ears over and over, and you can tell that there's a lot of differences. A lot of it is correlated to sugar, as you would expect, but not all. And if you think about boiling sweet corn and you get the corn aroma coming out of it, there are volatiles that are explaining some of the taste perception. And so what we're doing as part of this project is not relying on the breeders, which is a sample size of 1, and it's just I like it, and not necessarily you guys' perception would be equal to mine. So we're getting as many as we can of hybrids, and many of which are coming from crosses of the inbred lines that are genotyped, and putting them under sensory panels in the food science department. This is being led by Charles Sims at the University of Florida. So basically, we get about somewhere between 60 and 100 people to try the variety and rate several questions: how sweet it is, how crispy it is, how much you like it. And then I'm not going to show any data on here, but Paul Mitchell at University of Wisconsin is actually coming in and asking what's the willingness to pay for each of these varieties to see if flavor actually will correlate with willingness to pay. It's a very interesting assay where basically they actually give is a experimental auction, so you actually give money to the panelists, and they have the option to walk away with the money, or if they say that they're willing to pay X, they basically buy the corn and take corn home and give back the money or part of it, whatever they're willing to pay. So it's a real, at least as close as we can get. So we're doing this, and then for each of these lines, we quantify carbohydrates, sugar, and volatile profile. So this will generate a large data set that we hope to understand if volatiles are important, and if so, which ones are, how much sugar really affects perception. We're getting some really interesting evidences on texture and pericarp thickness and some other traits that breeders knew they were important, but we don't really know how much they are important relative to sugar. And we're doing a little bit of imaging processing, and again, that's where I wanted to, full disclaimer, this is very, very rudimentary, even very simple actually, but it's really the only way that I can, under constraints of a public program, to really scale up phenotyping. I mean, you come to a breeding program full of these theoretical things that we've learned on applying BLUPs and experimental designs and how to actually create the optimum design. And the reality is, a lot of the times, there's just several constraints, and specifically in the case of a public breeding program. So we're trying to go as simple as we can, because really for us, it would take a lot of time and effort to develop probably something a lot more complex. But the way we're dealing with image processing is mostly just dealing with segmentation and not so much about modeling. So we take pictures of all these ears and we segment the colors out, the different colors. In the case of ear length and ear width, this ends up becoming a 0-1 matrix. So that I just put it on Excel here, and you can see where the ear is just color-coded 0-1. And this becomes actually fairly straightforward to just count pixels and measure what's the year length and what's the year width. So we're now putting that into a tray where we can do several years at a time. And the next step would be then to collaborate with some folks in engineering to put that in a conveyor belt and just do that over and over again. Correlations, I'm sure they can get better, but as of right now, we're pretty happy with what we're observing. So we get about 0.93, 0.92 for the measurement of width and length correlated with the manual measurement, but it saves a ton of time to do that through pictures. And that really just highlights a little bit of what we were talking yesterday of the importance of phenomics. And I agree that I think phenotyping will be the frontier. So we're using the same tool to quantify tip fill, which is probably one of the very important traits for us. We're looking at color. And ultimately, the goal is just to have an automated system where we're getting a spreadsheet out with phenotypes that we can then move to the breeder. The only final example I wanted to show is that a similar approach for phenotyping of southern and northern corn leaf blight. Those are two diseases that are important down there. Incredibly, southern is actually becoming more important recently than northern because some of the fungicides apparently are not working as well in Florida. So we did an inoculation test. We inoculated the fungi in the diversity population. We have a significant amount of diversity. Heritabilities that were okay, that at least there is genetic variability there. But it really takes a lot of time training the students to do this rating and to go to the field and know how to score. So the approach we're taking is, again, using a lot of different segmentation, and I won't go into too many details, but we take pictures of these lines, we extract the barcode, segment out the background and the veins, and then end up with a disease leaf area. And note that here we have both southern and northern at the same time. And it turns out that this actually did increase the heritability. And I just yesterday ran a quick GWAS on these phenotypes with the data the genotyping data that we got. And it seems like we are getting some hits that we'll have now to explore. So that's something that's ongoing, but we're quite excited. We think it will at least accelerate the process. So I got 12 seconds left, so I think I'm good on time. There are several things that are going to go on as next steps. We're really trying to finish and figure out a lot of these genomic approaches, calibrate the models, identify a panel of 3,000 SNPs for imputation and application of genomic selection. We're collaborating with Alina Zaire from UF to now use it's a method that she and collaborators created. It's called multi-target, multiple instance adaptive cosine estimator that will allow us to separate in the picture what's southern to what's northern. Right now, we're just getting the disease leaf area. And I'll be happy to take any questions. This is my group now in Florida. This is a collaboration of a large amount of people. And thank you very much.
Fantastic. Thank you for your talk. So at this time, if anyone would like to come and ask questions, feel free to come up to the 2 microphones here. We would love to get some interesting conversations going.
Thank you for your talk. I just had a quick question about the sensory panel and how that plays into your decisions. In the beginning of your talk, you mentioned how you can get 3-4 cycles a year. And so the turnaround and your decisions must be extremely quick. But you're sending the sensory panel, or you're sending the samples to be processed by a different lab. So how does that play into your decisions when you're making selections so rapidly? And do you weight it, or is that just more of a secondary thought?
Great question. Thank you. So as of right now, the sensory panel is not really playing a role in any of the decisions of the breeding. We do a bite test in Idaho, and we do another bite test in Belglade. So on the fly, basically, the breeders will collect data. Now, I have a side project working on tomatoes where using metabolomics data, using sugar plus volatiles, we can actually predict very well the outcome of these sensory panels on average. So I can give a tomato to each one of you here and you will have a different perception, but on average, I can predict whether a given variety is better than the other. So at one point, the expectation is that if flavor becomes an important trait, we could quickly, the same way genomic selection works, we could quickly do a metabolomic selection, if you will, and select for these panels. Because they're not very high throughput. You cannot feed 300 varieties to a panel, you get tired of it. So you usually run 4 to 5 varieties per panel.
Thank you for your talk. I'm interested in the the chromosome 4 lack of diversity that you showed on the sweet cap. So what do you think is driving that? Is it a form-specific introgression? And I looked quickly at MAGE-GTB and didn't see shrunken 2, I don't think was in that chromosome. Neither was SU2. So do you think there's any sweet corn-specific introgression that's driving that lack of diversity in chromosome 4?
So sugary 1 is in chromosome 4, and shrunken 2 is not, but every shrunken 2 line was basically, shrunken 2 came in the 70s where there was a lot of sugary 1 breeding, so most of them are conversions. And this is a really good question that we're trying to understand. There is a gametophilic in there, TB1 is there, that we're trying to really understand what's going on, but there's clearly low allele frequency. There is a segregation distortion QTL there as well, and we don't really know exactly.
Thank you for your talk. So I'm a field breeder, or field corn breeder. So, obviously harvestable yield is by far the most important trait that you're breeding for, but it's really interesting hearing, the perspective from a sweet corn breeder. So maybe more of an elementary question, but I was just wondering, with yield being the end all be all, all these secondary traits that you're selecting for, are there negative correlation with those traits, or do you know the solid correlation between those?
Yep, thank you. For us, yield is definitely the most important too. We do have some of the constraints on the aesthetics that they have to be there to be commercial, but a lot of flavor is there is definitely a distinction on what goes on in the field breeding parts of my program and the research that maybe one day can be applicable if the trait becomes more relevant. We are looking into some of these correlations right now. I don't have an answer for you as of yet, but definitely yield is the main priority.
Awesome. Thank you very much. Let's give one more round of applause to Dr. Resende here. So really quick, we also really appreciate taking your time to come and travel and visit us. So as a token of our appreciation, we'd like to present you with a small gift and say thank you. So we're going to get the next speaker set up here for a second. And while we do that, if you have a morning poster, especially this morning, we need you to go and get those put up. That would be fantastic. So again, morning poster, if you want to do that, that would be great. So bear with us, have some great conversations. We're going to get set up for our next speaker here.